Hello everyone and welcome to the latest MDPI webinar on toxics, which is entitled Microplastics Distribution, Fate and Effects on the Environment. Thank you very much for joining us today. This is the third webinar of a series that is based on special issues and sections of our toxics journal. So if you wish to submit your paper to any of the special issues related to this webinar, please check the webinar's website. Our chair today is Professor Juan Bellas from the Spanish Institute of Oceanography. We will also have three speakers, Dr. Gilberto Binda from the laboratory of Professor Roberta Bettinetti at the University of Insubria in Italy, Professor José Manuel Andrade-Garda from University of A Coruña in Spain, and Professor at Ragas from Open University in Heerlen and Radboud University in Nijmegen, the Netherlands. At the end of the session, we will have a Q&A session during which we will address some of the questions submitted by live online audience. So if you're joining us live, please submit your questions at any time during the webinar in the Q&A box. And as we have more than one speaker today, please mention the name of the speaker together with your question. I would like to remind everyone that this webinar is being recorded and that the recording will be posted soon at the webinar's webpage. So now I'd like to introduce our chair. Juan Bellas has a PhD in marine sciences in the field of ecotoxicology and marine pollution. He's professor of research at the Spanish Institute of Oceanography. His main research focuses on the study of the biological effects of pollutants, including microplastics, and the integrated assessment of marine pollution. He's the chief investigator of the Marine Pollution Program of the Spanish Institute of Oceanography, and he was coordinator of the scientific activities for the implementation of the Marine Strategy Framework Directive in Spain from 2010 to 2018. He has participated in various international committees and working groups of the European Commission, groups of the OSPAR Commission, and of the International Council for the Protection of the Sea. He's an advisor to the Ministry of Ecological Transition of Spain and a member of the UNESCO Chair in Sustainable Littoral Development. He has participated in several oceanographic cruises, including 3 cruises in Antarctica, and participated in more than 30 projects and research contracts. He has published over 90 scientific papers and several book chapters, and he has been granted 3 patents. So now I will hand over the floor to our chair, Professor Juan Bellas, and all of you enjoy the webinar.
Thank you very much, Elena, for the introduction, and welcome everyone to this webinar organized by Toxics. First, I will introduce the journal that is organizing this webinar, which is Toxics. It's an international peer-reviewed open access journal that covers different aspects of the toxicity of chemicals and materials. The journal was indexed by the Science Citation Index Expanded by PubMed and Scopus. The scopes include a wide range of perspectives on the exposure of humans and the natural environment to toxicants, the effects of toxicants on human and ecosystem health, and methods to manage and mitigate these effects. The 2018 impact factor for toxics is 3.271. It ranks Q2 in the category of toxicology and environmental sciences. And regarding the processing time, the manuscripts are peer review, and a first decision is provided to authors approximately 15 days after submission. And acceptance to publication is undertaken in 2.8 days. So it's rather quick. You are also welcome to review for toxics. When reviewing, you will receive several benefits, such a discount to share and a personalized reviewer certificate. So as for this webinar, the title is Microplastics Distribution, Fate and Effects on the Environment. This is a very relevant topic, and that has received exceptional scientific attention over the last 10-15 years. Just to have an idea about the problem, since the decade of 1950s, the world production of plastic products has increased exponentially, reaching almost 360 million metric tons in 2018. And as a direct consequence of the industrial activity, consuming habits, poor waste management, and plastic wastes have been found all around the world in the environment. Due to their persistence in the environment, plastic pollution has become a large-scale phenomenon, sort of say, posing serious threat to the marine environment, where about 8 million tons of these materials are released annually. Microplastics, which are defined as plastic particles below 5 millimeters in diameter, include primary microplastics that are produced in a small size to be used in plastic production, cleaning, cosmetic, or medical products, and secondary microplastics resulting from the fragmentation of larger items in the environment. After a decade of intense research effort on this field, numerous studies have shown the widespread distribution of microplastics over the planet and their effects on the ecosystems. So, besides the distribution in the environment, issues such as the toxicity of plastic additives like antistatic agents, UV light stabilizers, flame retardant, plasticizers, fragrances, etc. The toxicity of these plastic additives have also been studied. Also, the ability of plastics to absorb toxic pollutants that are in the environment have been studied, and including the bioaccumulation of these microplastics on different levels, on the biota and different trophic levels. However, there is still a general lack of understanding on several aspects that need to be addressed. Some of these gaps of knowledge include the incorporation of microplastics and associated pollutants into food webs, the joint potential impacts of microplastics with other stressors in the environment, the lack of standardized sampling methodologies, or the distribution and the impact of the lowest particle sizes. So this webinar will discuss some of these current challenges in microplastic research, aiming to contribute to advance in these issues, to contribute to the assessment of the distribution, the fate, and the effects of microplastics in aquatic ecosystems. So this will be the focus of our talks. Essential speakers. Our first speaker is Gilberto Binda from the University of Insubria in Italy. He's a postdoctoral research that holds a PhD in environmental chemistry. And so with that, I will give the floor to Dr. Binda. He will talk about the issue of microplastic pollution in freshwater environments. So thank you very much, Gilberto. I give you the floor, and you can start with your presentation.
Thank you very much. I will start sharing the screen. Okay, then. Today I'm here talking to you about some effects, some things, and sources of microplastic, with main focus on water environments and more specifically on freshwater environments. I will talk today on behalf of Professor Dr. Roberta Bettinetti, which is an ecotoxicology working in Insubria University in Como, Italy. I will start with the key points which I will touch during today's speech. I will start with a brief introduction regarding history and how plastic start to be used so widely, and which will be basically the main causes of the extremely diffuse pollution which we are working on now. And then I will talk about the production and how the plastics start to move in the whole environment and where we can find it. I will focus then on microplastics and to see what are they, how they form in the environment, where we can find them. And then we will focus on the water bodies in more details and see their behavior, especially talking about the chemistry and how they interact with other chemicals in the water, and even with the organism and the possible toxicological effect, which still need to be studied, especially in freshwater environments. Finally, we show you some data, some first reports regarding an Italian case studies on which we are working on, with the main focus clearly on freshwater in this case, which would be Como Lake. And then we show you shortly some future projects which we are still working on, and we will try to better develop in the following period. So starting with the history of plastic, the history of plastic starts about in the second half of the 19th century. And in that period, there was basically a great finding, and even a green finding, which is pretty strange since today we are talking about plastic pollution. But in that period, plastic, which was great material, easy to mold and easy to prepare and very durable, was the substitute for some object, likely some jewelry object or a ball billiard, was the substitute of ivory, avoiding a very bad environmental problem, which is the collection of ivory. In more detail, the first polymer which substitutes ivory is called bakelite and was synthesized in the beginning of the 20th century. Then, moving through the 20th century, plastics start to be more widely used. Different other polymers start to be discovered and used. You can see in this slide there are some polymers which are pretty famous by now, likely polyvinyl chloride, nylon, PET, and starting from the 60s, plastics begins to be used by the whole population, and the use of plastic continuously increased and became very popular also for different use, from the industrial use to the home use with families. So now I will show you some details about the production of plastic, which is called polymerization, starting from which are the precursor materials of these polymers. Even if we are recently try to move in the direction of the bioplastics, so plastic which can be produced by biological material, for example, the rest of some biomasses like castor oils or even bioethanol. But so far, still, most of the plastic polymers are still produced in the synthetic way, so starting from the extraction of crude oil. I will show you in this slide in a bit more detail the production of plastic for synthesis, starting from the extraction of crude oil, which is a pretty complex mixture of different hydrocarbons, which are used for different applications, which are then moving in the refinery plants. Oil is fractionated to the distillation, and some phases, naphtha is the most used phases for the production of polymers. In this phase, naphtha is separated from the other fraction of crude oil. Then plastic is produced, starting generally from the monomer. In this example, in the slide, I put on ethylene. And this process is called cracking process. So we obtain the purified monomer composite, which finally is finally is produced with the polymerization, so linking all the monomers together. We can have the polymer pellets, which generate then and are molded to the instrumentation or the various different objects made of plastics. Moving a bit on the properties of plastic, which are which make this material very widely used. In fact, each polymer can have different properties, which clearly depends on the chemistry of the single monomer which is used. But in the years, different additives were added to improve different properties, especially mechanical properties or physical properties like flame resistance or more elasticity, were added to even improve these properties of plastic. But generally, plastic became pretty famous because it presents a low cost and an easy for the production. It is generally a very durable material and pretty strong, but even light, not very heavy. Moreover, it has some other peculiar characteristic, likely electrical or thermal resistance and resistance for mechanic stress. And as in this way, as you can imagine, plastic start to substitute different other materials, both for industrial and for home use as well. And even if basically a lot of polymer materials are registered in the European Union, you can see from the slide that we have about in Europe, we have about 30,000 polymer materials registered, but actually most of the plastic use is made in really few polymers. In this slide, I reported 4 pictures of pretty different materials, starting from tubes, moving to bottles, bags, and another material which became pretty famous in the last year, which is the face mask for protecting from different likely from the COVID-19, and all these materials are made of polyethylene. So as you can see, one single polymer can be molded in very different ways. And this is another key point for the whole big production of plastic. All these fantastic properties turn in a wide and abundant use of plastic. In the picture on the left, you can see the increasing trend from the 50s until today, which is indicated in red. And then the forecast globally even shows that possibly plastic use could be even more increasingly in the following years, even if policies start to move in order to try to at least produce some more easily degrading plastic and other more green materials instead of the one which are using now. And this wide use turns into a global problem regarding the environmental fate of plastic. In fact, the very important and famous properties of plastic, one first of all is the high mechanical resistance, make this material really durable even once dispersed in the environment. And therefore, this good property became a problem for plastic pollution. In the recent year, in fact, plastic is reported to be found basically in every environmental compartment. It was found from the glacier and the mountain in the Alps to the sea, and in different water bodies. It was found moving even through the atmosphere as a particulate matter. It was found deposed in soils and sediment. It was found inside animals, and therefore it was found basically everywhere. But a very hit environment by plastic was a water environment. In fact, some even different models find out that most of plastic finish as a final sink in water bodies, and recent estimations indicate about 8 million tons of plastic which enter in oceans for every year. You can imagine this is going to be quite a big environmental issue and need to be solved. Especially because once dispersed in the whole environment and different environmental compartments, human as other animals interact with the surrounding environment, and plastic, once dispersed, can come finally back to us. In fact, as shown in the picture in the right, there is a little sketch regarding some way of intake of plastic for human beings, starts likely from the ingestion of microplastic in aquatic biota, like fishes or like mussels, and the problem of microplastic in drinking water, and even found in other foods, likely table salt. In fact, some recent research find out that microplastic was even found in some human organ and even in human placenta. In this presentation, I will mainly focus on the microplastics, and so the fraction in size regarding the different categories which are described in literature. Microplastic indicates all the plastic which present a dimension which is between 5 millimeters and 1 micrometer. These plastics can be of generally different shape, and can be of different type. Categorizing them by their source, we have this. We have the primary microplastics, which are microplastic which are already produced as in the micro size and then dispersed, typically inside cosmetics. And secondary microplastics, which can result from the breakdown of larger microplastic in the environment. And while regarding their morphology, we have a wide variety of plastic materials. As we have different plastic materials, we have different morphology in the final plastic we find in the environment. This issue make even harder their sampling as well as their analysis in environmental samples. I will focus now on main sources and sinks of microplastic in water bodies. I will try to focus a bit more on freshwater environment, but more knowledge is related to seas, and then we'll start from seas and then move into the freshwater environments. One thing that is start to be clear is that quite a big portion of microplastic found in waters actually have a land-based source. So some problem related likely with public littering, waste mismanagement, area highly touristic areas and industrial activity can have problems, especially if fresh water, in this case, fresh water is not treated rightly, likely in a wastewater plant. And therefore, the land is the main source, but then usually plastic move to the water. There are, in this slide, I show you some data, and then we try to figure out who to blame for microplastic presence in water bodies. In fact, we can see that 80% of plastic particles are released in fresh water from likely industrial waste. Can we consider that the whole fault is of the industries? No, actually, in fact, a lot of fibers, most of the fibers found in freshwater environments are related from the discharge of domestic washing machines. And as well, most of plastic particles, primary plastic particles, come from different cosmetic products. Can we blame then some efficiency of sewage plants? Actually, neither, because it is reported that sewage plants can retain almost 99% of the particles found in the water bodies. The main problem is that actually there are a lot of plastic to manage. The amount is really abundant, and even a very slight portion directly dispersed in the environment can cause several environmental issues. Just a slight overview on cosmetic products, which is probably the first source of microplastic, primary microplastic in some freshwater bodies. But another big issue is that once we know we have this problem, it's not easy to regulate to policy these kind of materials. For an example, in Italy, the use of microplastic is extended to be rinsed and prohibited from the beginning of 2020, but not all cosmetic products fall in this category regarding their legal use, and in this way, microplastics are still used in some products. I will move now, starting from the sources, and I will move now to the sinks. The sinks with, of course, a main focus on water bodies. In this sketch, you can see, as we already presented before, that main sources of microplastic in waters are due to land-based sources, but we have also some deposition directly from the water, likely from boat, from fisheries, and even the long movement to atmospheric deposition. And then once in water, microplastic can sink or can float depending on the polymer density, but then their behavior in water can even change during a process called plastic weathering. Moreover, throughout this movement through the water column, plastic can interact with biota, or can be ingested, can sink, can be fooled by microbiota, and can even then be weathered to form smaller particles, or even accumulate in the sediments. I will focus now in more detail on the plastic weathering, since this phenomenon seems to be really important, especially regarding the chemical behavior of plastic once dispersed in water. As we already stated before, microplastic, once fragmented in the case of secondary microplastic, or primary or immediately dispersed as micro in the case of primary microplastic, undergoes some physical and chemical processes, likely the receivement of solar radiation, some oxidation processes, and can be colonized by microorganisms. All these phenomena permit to change some changes in the surface activity of plastic, even in some physical and chemical properties. For example, the colonization make the growing of some biofilm on plastic. We have some disgregation and then some change in porosity and surface properties of plastic, and some usually change in crystallinity. The amorphous region of the polymers seems to increment with long weathering. Moreover, the oxidation processes permit the enrichment of plastic with some oxygen-containing groups, of course, changing their chemical reactivity. Moreover, all this phenomenon can cause some changes even in density of the particle. Once the particle changes its density, it was observed in different studies that, for example, the biofouling of plastic and their weathering can form denser particles, which can, instead of float, start to sink. And once these particles sink, usually accumulate in sediment, in the sediment compartment. This compartment needs to be carefully addressed regarding the possible effect of microplastic accumulation here, because if an high amount of plastic will accumulate here, this environment presents some peculiar condition, likely some reductive chemical conditions, the abundant background of some compounds and contaminants which are usually abundant in this environment, likely some hydrophobic contaminants or trace metals, and therefore this environment in detail need to be better addressed regarding even experimental conditions, as well as possible negative effect which can happen on the benthonic fauna. Talking about the interaction of fauna with microplastic, these particles, which can be pretty little, as we already told, regarding the dimensions, can easily be ingested, even by some small organisms, and can have different direct physical effect, likely the reduce of feeding rate and some negative effect on the ecophysiological functions of the animals. But even if they have some changes regarding their surface chemistry, can introduce some other chemicals to the food web, and can therefore behave as a vector of contaminants. And in this case, of which contaminants are we talking about? We are talking about some additives or even some adsorbed chemicals which can interact with microplastic along their weathering in water bodies. Focusing firstly on additives, plastic is made with some as told before, plastic is made of some polymers, but to improve some physical and chemical properties, often some other chemicals are added to these polymers once we prepare the final plastic product. Some example of them are likely plasticizers or flame retardants. These chemicals, anyhow, have possibly some toxic and negative effect once leached from the plastic particles. And in the case of plastic weathering, likely oxidation of plastic surface can easily cause this release, and another peculiar environment, which is likely the gastrointestinal tract, can even enhance this leaching. Moreover, in a few cases, some monomers, so just single compound of plastic polymers, can be leached from plastic after their weathering, and some monomers are toxic as they are, so can have other negative effect regarding toxicity. Moreover, as already told before, some toxic chemicals can interact with plastic particles and can present problem if plastic is ingested. It is recently known that persistent organic pollutants as a lipophilic component easily interact with plastic particles, since they're chemically pretty similar, and lipophilic component can easily be absorbed on plastic polymers. In this way, possibly plastic, once ingested, can cause the release of these chemicals. Moreover, pretty recently, some studies, some reports, and some experimental some experiment on simulated water environment observed that plastic seems to interact even with some trace metals, which are pretty toxic in some cases as well. This is pretty surprising because ionic species seem to not to be absorbed on lipophilic compound, but it seems that plastic aging, especially biofueling, can play an important role even on this absorption. And this issue start to be highly studied in research. To show you here an example regarding the absorption of a persistent organic pollutant on plastic particles. In this slide, there is a global map regarding the concentration of DDT, which is a pretty toxic compound found on plastic, absorbed on plastic pellets in different areas on the different oceans. And while there is clearly high variances, for example, we have some hotspots on the East Asia and on the west coast of the United States, but globally it was found to be absorbed in all the area where it was sampled. These kind of issues still need to be completely studied in fresh waters because we have not as much data as in seawaters regarding these effects. Another effect which still needs to be validated and observed in freshwater as well as in seawater is the possible effect of biomagnification of contaminants driven by the ingestion of microplastic. It should be still validated if plastic can more easily cause a biomagnification effect and therefore an increasing concentration moving up on the food chain, starting from the prey, moving to the predators. I will focus now on freshwater bodies in more detail. In the graph that you can see on the left, there are some number of studies published in 2019 regarding microplastic in water, in the biota, and in sediment, with regard to the difference between marine water, which is indicated in blue, and freshwater, which are indicated in green. As you can see, freshwater are not as studied as marine water regarding this environmental issue, but freshwater present an important societal and environmental role regarding, for example, the human consumption of the water human consumption and the industrial use of fresh water. And moreover, fresh water can be considered as the road to the sea for land-based source of plastic. Therefore, the way plastic moves through these kind of water bodies is extremely important to even understand also how it can finish in the seas. I will focus now in more detail on the case studies on which we are working on in the University of Insubria, which is Lake Como, located in northern Italy. It is the third biggest lake in Italy, just beside the Alps, and this lake presents some important societal and ecosystem function. In fact, there are some fisheries. As a big lake, it is used as a water source for different human applications, such as agriculture. And moreover, the plastic pollution here can have some important even societal repercussions because this area is a highly touristic area. Show you fastly some recent report regarding the presence of microplastic in Lake Como in 2018. A preliminary survey of floating microplastic in this lake was performed in collaboration with an environmental society in Italy, which is Legambiente. And what was found is that about a bit less than 30,000 microplastic particles per kilometer squared were found floating on the surface of Lake Como. In the slide, I also reported some other closed lake, like the Garda Lake, which is another Italian lake, the Rhine River, and the Constance Lake. And we can see that the abundance in terms of particles are pretty similar with Lake Como ones. I show you also here a recent study which will be published by Professor Bettinetti, in which the presence of microplastic was quantified inside a predator fish, which is the common perch in Lake Como too. In these studies, the main finding include that most of the sample fishes, so 87% of the fishes, present microplastic pellets in their tissues. I will finally show you which trend will be included in the next studies in this study area, what we are working on. We are moving for the quantification of microplastic, focusing on the sediment of Lake Como, try to better identify how the sources and the transport mechanism can affect the distribution of these materials in our lake. And we will move further for the evaluation of the possible interaction between microplastic and organism, with the main focus on the bentonic fauna, once again to move into sediment. And another research branch which is pretty moving is the understanding of the possible interaction with other contaminants, both organic and metals, which are known to be already present in Lake Como. Now I concluded with my presentation, and I will thank you all for your attention.
Thank you very much, Gilberto, for this very interesting view of the issue of microplastic pollution, with focusing on freshwater environments, which was very excellent presentation.
Thank you.
Okay, so now we have the second talk today that will be given by Dr. José Manuel Andrade-Garda. Dr. Andrade is professor of analytical chemistry at the University of A Coruña in Spain since 2011. His current interests are the infrared analysis of microplastics, FTIR applications to accelerate industrial quality control, and data mining. And he also works in petrochemical and natural gas analysis. He participated in dedicated training courses at several industrial laboratories and various universities. So now I give the floor to Professor Andrade, who will talk about the measurement of microplastics by mid-IR spectrometry. So José Manuel, please, you have the floor.
Hello, Juan, everybody. So I will start. And here you can see, just me check here. Okay, here you can see the three main parts of the talk. First, we will discuss something about environmental matrices, not in the way that Gilberto made, but just talking about analytical problems with sample matrix. Then we will go into characterization, and finally, we will make some conclusions. When we talk about environmental matrices, this is just not to explain you about the problems of water, air, etc., because this was very well addressed by Gilberto, but to remark that only very few times you will get easily the specimens, in this case the microplastics, you want to study. In many, many cases, you will need to extract the specimens from the matrix. This is an example from a beach, and you can see a lot of specimens that can be picked up very, very easily, but this is not the case in water, or this is not the case in sediments in many, many cases. So let's suppose that we will work with water. Well, if your water has a very few content of organic matrix, then probably you will be obtaining a very good work if you just filter the water, and the remains, the filtering, the filtrate, you can just wash it with ethanol or methanol, or much better with hydrogen peroxide, to avoid the organic remains that you could have onto the specimens. On the contrary, if you have moderate organic contents, you need to be more drastic in the sense that you need to get rid of the organic matter. You can do that many, many times by hydrogen peroxide digestion, typically 24 hours. Many, many authors suggest to include a pre-digestion using a surfactant like Triton or SDS or any other. This is just to simplify the work. This is an example. The picture at the left shows you the filter after filtering the sample, before the digestion, in this case with hydrogen peroxide. And after the digestion, you can see that the filter is really, really clean. The organic matter has been digested, it disappears, and then you can identify more or less easily the specimens, the microparticles. If you have strong organic contents, like for instance in wastewaters, probably you will have a lot of troubles. You have a complex situation, and probably you have to apply a digestion which is very, very time consuming and very, very lengthy, and of course expensive, unfortunately. In fact, many, many times you will apply a protocol which has been developed originally by US NOAA, and that was modified quite recently by Gunnar Gertz from Germany and Jess Wollertsen from Denmark. And in fact, you can see here a resume of the procedure they suggested. It is a very good procedure in the sense that the microplastics are not affected by the digestion, but you can see that it takes more than 1 week per sample. Okay, maybe you can process several samples at the same time, but probably this is not very convenient if you are performing monitoring studies. You know that in monitoring studies, we get a lot of samples. So you can see that you need a huge amount of work, a lot of money, because this is quite expensive. And of course, you have to be patient because you will need a lot of time. If you are interested on wastewater treatments or any kind of water treatments, you can take a look on this review, which I think it's quite nice and very, very recent. Let's suppose that we are working with biota. In biota, when you review the literature, you can find at least 4 main approaches. And I am saying just at least because there are many approaches, but I think these are the 4 main ones. You can perform an acid digestion using a typical strong acid like hydrochloric acid or nitric acid. Well, in general, I should not recommend this option. And the reason is that these strong acids, even when diluted, they can degrade a little bit in microplastics. So I would suggest you not to use this approach if you can avoid it. You can use exactly the opposite situation. You can use an alkaline pH like sodium hydroxide or potassium hydroxide. Just be a bit careful with the concentrations, because especially sodium hydroxide tend to affect also the surface of the polymers. Enzymatic treatment is very, very common. You can find many treatments with different enzymes, different concentrations, even mixtures of enzymes. And in fact, the treatment of the wastewater I showed you before is based on an enzymatic treatment. And the reason is, okay, enzymes preserve quite well the microplastics, but they need time, and some of them are really, really expensive. You can perform also an oxidative treatment, typically with hydrogen peroxide or hydrogen peroxide combined with iron in the state of 2, just because it is a good catalyst. And in general, this oxidation is a complement of the previous ones, but I have to say that many, many times this option is good enough just to get a treatment of the sample from biota. You can see that I am telling you very, very general ideas, And this is a problem, and this is not because I am just doing that on purpose. The reason is that every matrix is different. If we talk about fish, in fact, we should specify much more, because if I am talking about salmon, which has a lot of fat, it is not the same as cod or hake or any other typical fish without so much fat. So the treatment in many cases has to be optimized for your particular matrix. And so this is why you can find a lot of protocols, a lot of different methodologies. This is just an example we made here in our laboratory. We wanted to treat fish stomachs and mussels, and we performed a very first treatment with a surfactant, sodium dodecyl sulfate. Then we adjust pH to add protease. We made another treatment with lipase, and finally we clarify the solutions with hydrogen peroxide. And you can see that the filters at the end are really, well, at least acceptably well. They don't have very big remains of organic matter, and they are quite clean. So this could be useful for more for subsequent steps. What happens if you have to deal with sediments? Well, this is a complex situation. First, you have to float the microplastics from the, or at least microplastics and other suspicious particles from the inorganic matrix. For this, you need a flotation solution, and there are many options in literature. The typical one, because it's very cheap, is saturated solution of sodium chloride. This is good enough if you are interested on light polymers like polypropylene, like high density polyethylene or polyamide, for instance. But it is not good if you are interested on the heaviest polymers, like, for instance, PVC or polycarbonate. For this, you need solutions typically based on tungstate solutions, but you can also use, you can see here the tables, sodium iodide or zinc chloride, for instance, which have more density. There is another option, which is to use elutriation. Elutriation has been used from the 60s, from 1973, typically by biologists, to separate different kinds of organic matter. They use an upwards flow, typically of water, but here for microplastics, you have to combine a flow of water with bubbles, with air. And here at the top, you put the sediments. Of course, sediments tend to settle down, so they tend to go down. But due to the upward flow of water and bubbles, then they separate according to the density, according to the size, according to the flotation properties, etc. And at the top, you will obtain the finest particles, could be inorganic, of course, but also, hopefully at least, microplastics. This procedure was presented first time in 2013, and it was a little bit refined in 2018. And well, it is a bit costly. It takes about 4,000 euros at least. They are newest systems. You can find them if you look for the papers from Gunnar Gertz, but the new systems are even a bit more expensive. So maybe you have to balance. This is the problem you can find with you are working with sediments. In many cases, you need a lot of filters per sample, let's say 8, 10 filters per sample. And this has been reported in literature because, as I told you, you are not only floating the microplastics, unfortunately, you are floating the finest particles, which are inorganic as well. So you will have a lot of fine particles in the upper surface, and so you will find them into the filters. Of course, if you are lucky, and your sediments have a lot of stones and big particles, when you make the flotation, you will obtain a nice result, of course, like this is the case. But if you are working with sediments like Galician sediments at the north of Spain, they are very muddy, so you will obtain a lot of fine particles. So in order not to get here a real block of mud here, you need to filter through different filters. So this is not very convenient. In a project we are collaborating with, which is an European project called Microplastics, we hope that we can solve some of these problems and especially explode the new ideas for field studies, because I repeat, in field studies we have a lot of samples, so this is not very convenient. So let's go to characterization. In characterization, we try to objectively identify the main polymer that constitutes the microplastic, and of course, this is trivial, but this is clear that you also have to differentiate microplastics from inorganic or from any other organic kind of particles, which unfortunately they are present there into the filters. This is a first idea I want you to keep on mind, please. Microplastics evolve. This is something that Gilberto said in a slide. Yes, it is unfortunately very, very true. Microplastics polymers evolve when they are in the environment, especially they photo-oxidize. They have more transformations, but this is a very critical one. So if the functional groups move, they change, then the infrared spectra will change. And this is critical because most identifications nowadays are based on comparisons between the original spectra you obtain in your system and commercial databases. And in general, commercial databases don't have weathered spectra of polymers. So you have to be very, very careful, because otherwise you can think that you are visualizing a very young lady, but in fact you can see an old lady, or you can think that you can see only an old lady, but in fact the young lady is there. In fact, both are there. So be very careful with your identifications. This is very, very critical. This is just an example from our own laboratory. The red spectrum is the typical one, the very nice one you can obtain in any book, in any paper, in any database, and this is really, really nice. When you obtain a black spectrum like this one from a particle, initially you would never think that this is low density polyethylene, which in fact it is. But, and we also had the same problem until we realized that polyethylene degrades in nature. And according to Gardet and co-workers, this is the mechanism for the degradation of a polyethylene. And when you study this mechanism, you can justify all the new spectral bands. And you can see that the spectra are really, really different. But in fact, they respond to the same polymer. So again, please be careful when you make identifications. We were very concerned by this problem, and we really made a deeper study and more extensive. We participated also in a European project, which was called Baseman, and we realized that for many, many field samples, when you challenge the common libraries with your original, with your real spectra, then in many, many cases, you obtain no matches. This means that the particles are unknown for your library. And once we feed the commercial library with our own database, and this was obtained using controlled weathering of different polymers, then the identifications were really, really good. In this case, it was polystyrene. And this is not the typical nice, very precious spectrum of polystyrene. In fact, we realized that this happens to about 70% of the polystyrene particles that we finally identified in our samples. So this is a big, big problem. Okay, how can we obtain the spectra? Well, I would say that this depends a little bit on the kind of particles you obtain in your filter. If you have big particles, I would say to you, be very, very simple. Don't worry, just apply common techniques, which are very common, very fast, they are simple, and they will not give you any kind of troubles. For instance, if you have, let's say, particles about 2, 3, 4, 5 millimeters, you can just put them on place on the top of a macro ATR, which is attenuated total reflectance, and then you can obtain this nice spectrum. It's not very nice, but it is not so bad. However, if you clean the particle with, let's say, methanol, ethanol, or just hydrogen peroxide diluted, then you obtain a really, really nice spectrum. And this is just a question of get rid of the dust of the surface or some kind of organic remains. If you have pieces which are like spheres or like squares, etc., you can also make another trick, which is just to cut the piece, and then you can measure the center of the piece. And the reason is that weathering occurs at the surface, not at the very, very center of the pieces. You can see here in a spectrum before cutting the piece. It's not bad, but it is not good enough. But when you cut the piece and you register the center of the piece, you get a very, very nice spectrum. If your samples are essentially laminar, don't worry, you can scrape off the surface and you will obtain a very nice result as well. If you have small particles, which is also very common, then you have to go to microscopes. This is very, very clear. This is an infrared microscope from Pelkinerma, And in any microscope, you have three different options to work. The first one is to use attenuated total reflectance, but in a micro scale. In our experience, this is not very, very good. It is not a very bad technique, but it is not good enough because you need to clean up the system every time you make a measurement. This is also typical of any other system, but here the system is a bit hidden. It's here, and you have to be very careful not to move the optical system, not to affect the optical system, etc. If you have 100 samples, okay, you have, 100 particles, you have to clean these 100 times, and this is not very convenient. Of course, you need to locate exactly the particle. It's not very difficult if your particles are big, let's say greater than 100 microns, but if your particles are very small, then, well, it's not so easy to locate the particle because you have to put the tip of the crystal on top of the particle. From time to time, you have to realign the systems, the optical and the infrared systems, because with the use they move slightly, not very much, but a little bit. This takes 2 or 3 hours. It's not very difficult, but time consuming. And this is the biggest problem. You know that most particles are rigid. So when you try to put the top of a crystal here on top of them, they move, they escape, and you will be angry because you will not be able to measure your particle. You will try to find the particle again, you will try to put the crystal on top of it, the particle will escape again, and at the end you will be absolutely crazy, absolutely angry. Transmittance is another possibility, and this is a good possibility, and in fact it is the best one if you can apply it, because you can obtain the maximum energy of your system, because the energy goes from the source to the detector. So yes, you can obtain here the best spectra. However, if you have here the particles, then you need the filter, of course, to be transparent. And this is not so easy. There are not so many into the market. Anodiscs are a possibility, and they are really expensive, so you have to think twice. But you can also use metallic filters, metallic mesh, which are a bit more cheaper. Not very much, but a little bit. Another thing, and this is an example, well, this picture and this one, you can find them in application notes from the website of PreKing-Elmer. They are free. Here you can see a metallic mesh with the particles on top of them. So in this case, the radiation goes from the bottom to the top. Remember that in spectroscopy, we have to obey the Lambert-Beers law, and they depend on the thickness of the particle. So if you have thick particles, maybe transmittance is not the best option. Okay, you have the maximum radiation of the system, but the thickness is so big that at the end you are losing energy. And this is the third option, which is reflectance, and this is the option we like most. The question is that you avoid many of the problems I have mentioned, but of course there is always problem in science, and the problem with reflectance, and this is just because of the nature of the technique, is that the signals are much weaker than, for instance, if you have transmittance measurements. Another question is that if you perform reflectance, you have to think a little bit before making the measurement. Maybe 30 seconds, not really more, but you have to think a little bit. You have to visualize the particle. If the particle has some approximately flat surface, and you see that the particle is not too rough, then maybe you can assume that the behavior of the light will be specular reflectance, like this one here. However, if you cannot find a flat surface, if you see that the particle is really, really rough, then quite probably the behavior of the radiation will be a diffuse reflectance. This is important, and you will see why in some seconds. So if you can select a flat part of the particle, then make the measurements there. Otherwise, I would suggest you to make 2 or 3 measurements at different sites of the particle and just look for the best spectrum. If you apply reflectance, you need to make mathematical signal treatments, and this is critical because the spectra you will obtain are really, really strange. So if you thought initially that your particle has a diffuse behavior, then you have to apply something which is called Kubel-Kamun. Should be implemented in the software, so don't worry, you don't have to make calculations by hand, fortunately. If you think that the behavior is specular, then you have to apply something which is Kramer's chronic correction. It is critical to know which of these 2 corrections apply, and you have to remember that because in general, you will not find commercial databases prepared for these two transformations. The result of the Kramer's chronic correction is very, very similar to an absorbance spectrum. So in many cases, you can apply still the commercial libraries, but if you apply Kubelka-Munk, you have to be careful. In many, many occasions, you will not be able to apply commercial databases because they are not prepared for this kind of transformations. Here you have typical spectra from reflectance. These are quite useless by eye. You need mathematical transformation. What happens if you apply the same transformations to both particles? The black one corresponds to an approximate specular reflectance from a nice pellet. And the red one corresponds to a diffuse behavior from a very, very rough and a very strange particle. If you apply Kubelka-Munk, you can see that this is nothing, but this is a very good spectrum because you are applying the correct correction to the correct original spectra. If you apply Kramer's chronic to both spectra, this is a very good solution because you are applying the correct correction for the correct spectrum, but the red one is totally useless because it's not good enough. So keep an eye always on the kind of particles you have and the kind of corrections you have to apply. And now we can go to the cinema. And if you can find this movie, I really recommend it. It's Hamlet from Sir Laurence Olivier, one of the most famous actors in UK. To be or not to be, to measure or not to measure the filter. Well, maybe you think that I am stupid because this question is stupid. But microscopes, nowadays, they are in the market and they can only measure an area of approximately 10 times 10 millimeters. And of course, most common filters are approximately 5 centimeters diameter. So it is clear that you will not be able to measure the whole filter with just one measurement. So you need to make a dramatic decision making. Will you measure one spot, 2, 3, 4, in which direction? Maybe you think, okay, I will make several measurements randomly. Okay, this is an approach that was studied in chemosphere in 2020. Or maybe you prefer regular patterns, which seems a bit more reasonable. There are different options. They were studied in analytical bioanalytical chemistry in 2018. Or maybe you think, okay, I will go directly to the central part of the filter. It's much simpler, and the spectrum is, sorry, the microscope is more easily placed. Okay, this was also made here in Nature in 2020. But I have very bad news for all of you. Regardless of the option you select, your results will not be good. And here, here you can see the reason. These are just 3 pictures from 3 different people. In this case, most of the particles became at the border of the filter. So if you measure at the center, you will not get good results. Here, if you measure at the center, you have a very good approach for the center. But if you extrapolate, you can see that you will not be in the right numbers. And here, it is a very typical situation. It is full of particles. It is crowded. Maybe you can see that one half of the filter is more crowded than the other. So if you make your measurements here and you extrapolate, you will get a total different result if you make your measurements here and you extrapolate. And remember, very, very few times you will be able to scan all the big filters. This is the reason why many people is going to 13 millimeters diameter filters. But in this case, you have to adapt all your sample treatment, all the amount of samples, all the amount of reagents, etc., and even the amount of aliquots you take. Because if you get a filter like this, or even worse, you will spend hours and hours or days, and probably you will not get a very, very good result. And remember, unfortunately, we don't have still unified protocols. This is very time-consuming and very complicated. We adopted an intermediate solution. We knew that with a little bit of experience, just by binocular microscope, a typical binocular microscope you can have in your laboratories, it is quite easy to identify the particles. At least if they are a little big, let's say more than 70 microns. Then we can pick up each suspicious particle, and then we place each particle in a special well that we prepare. This is a plate prepared in our laboratory. It is an aluminum plate with a lot of wells, and for each well, we locate one particle. Okay, yes, it is true that we spend some time here, but here at the infrared microscope, we save a lot of time. So combining these two options, we realized that we saved a lot of time. So, well, yes, I know you are thinking that what happens if I have particles less than, let's say, 50 microns? Well, we will not be able to catch them. But for particles bigger than 70 microns, we can catch the particles without big problems. And indeed, some 2 years ago, I would say, the main objective of this kind of studies was to monitor microplastics in seawater, for instance. And you know that in monitoring, we have to use manta nets. And manta nets usually have mesh sizes of about 330 microns. So in many, many occasions, if you are involved in monitoring campaigns, you will have to deal with big particles. So this is a good approach for big particles. Okay, I know that you are thinking, which kind of filter should I use? And this is a very, very, very complex question. I found a paper which I really like a lot, which is from 2020. And it is good enough for me, at least, not only because it studies different filters at different conditions, but because they considered field studies. There were many studies considering laboratory conditions for filters. But what happens with field studies? Well, they consider different conditions, different requirements, different limitations. And you can see that they recommended filters of 20 microns mesh size, and preferably of stainless steel. This is good because they are not very, very expensive. You can also use nylon or Teflon. They are also quite acceptable. An interesting point is that although the retention of very, very small microparticles are not quantitative, some of them still are at the filter, so you can make a qualitative study of minor particles. Well, at least this is something. But for particles which are, let's say, higher than 20 microns, you will obtain very, very high recoveries, which is what it is important. Okay, let's go to the conclusions. And you will see that my conclusion is a bit surprising. It is a pledge, and you will understand why. If I tell you that when you prepare your papers, you have to be careful and give all the experimental results, you would say me, okay, boy, this is elemental. Unfortunately, this is not the case in many, many cases. For the Baseman project, we needed a lot of information from infrared measurements, and we were surprised that we could not find information into the papers. And we realized that that was the general situation, which is totally crazy. So we took 100 papers and we studied the information they gave about the infrared conditions. And you can see absolutely astonishing results. Okay, 3 people out of 100 forgot to tell the brand and the model. Well, this is not really critical. Don't worry, this is not a big problem. But let's go to serious points. 83% of the people said nothing about background. And infrared spectroscopy, this is totally crazy, because in infrared spectroscopy, you have to take into account the background, and the background evolves with time. So if you don't take a background into account, you could have stupid results. Even more, 92% of the people said nothing about the periodicity of the background. And as I told you, this is critical. But even worse, 70% of the people said nothing about the statistical correction of the spectra. So I cannot be confident on 70% of the papers, because if they don't make the statistical treatment correctly, the results cannot be used to go to a commercial library, or the commercial library is not good enough. So please be very careful. This is absolutely critical. Unfortunately, this is something that it is happening in many branches of science. So I went on to different sciences like biochemistry, like PCR studies, medical studies, and they developed something which is called minimum information for publication of PCR studies, medical studies, etc. So I prepared, you can see here this little paragraph. This paragraph is here in this paper. You can just copy, and you only need to change these data, which are blank, for your own data. And everybody, you yourself, will be able to reproduce the study. Otherwise, it's really, really impossible. Please take this into account. And remember that in microplastic analysis, sometimes what you see, unfortunately, is not what you get. You have to be patient. You need a lot of time, a lot of patience. And remember that patience is the mother of science. And in science, not everything is disclosed to us first time. In this nice picture, you can see a young lady, but many people can see a very nice black panther here and a bird here, and both are there. So you have to take account of the problems you could have. Thank you very much to all of you. Thank you to Juan Bellas, and thank you to all the Toxis editorial team. And please, all of you, keep safe. And thank you very much.
Thank you very much, José Manuel, for this excellent talk and a very interesting presentation about a topic that is very relevant within the field of microplastic research. So now we continue. The third speaker today is Dr. Ragas. Ragas studied biology and obtained his PhD at the Radboud University of Nijmegen, the Netherlands. He currently holds a position as a full professor in environmental natural science at the Open University in Heerlen, and has an associate professor at the Radboud University in Nijmegen. His main expertise is the modeling of human and ecological risk of chemicals, with a focus on chemicals of emerging concern, such as microplastics. So now, for the last talk, I give the floor to Professor Ragas, and that he will talk about modeling microplastics from sources to effects. So, Professor Ragas, please, you have the floor.
Okay, thank you, Juan. First share my presentation. I hope it's visible now. So welcome everybody, and thanks Juan for introducing me, and thanks for MDPI for inviting me to this webinar. So I'm going to talk about modeling microplastics from sources to effects, and I think the nature of my talk will be a bit different from the other talks, because I'm not an experimental researcher, but I'm a modeler. That's quite a different perspective on the problem. So what am I going to talk about? First of all, I want to explain why I think modeling is important, and I do that by explaining the DIPSA chain. Then I want to go a little bit in depth about modeling exposure to microplastics, discussing the sources, the fate, and also the human exposure. And although I'm not an expert in effect modeling, I also want to make some remarks about the effects of microplastics and what we know about that. And finally, I want to make some short remarks about interventions and about how to implement intervention options in practice. And then I want to conclude with some conclusions and recommendations. So my first question is basically, why is modeling important? And I want to illustrate that with an example on six-pack rings in the environment, because I think mapping an environmental problem to understand how it is structured is very important. And one way to do that is with the help of the DIPSA chain. Many of you might already know what the DIPSA is, but for those who do not know it, I'll give just a quick example, and that's based on the pollution with six-pack rings in the environment. And there the problem essentially starts with that we would like to take a drink outside, for example, during a picnic. And of course, there are clever producers that think, okay, if you want to drink outside, it may be handy to put some drinks in a can, and we want to package these cans, and therefore we produce six-pack rings. So we produce them, and when you actually start using them, of course, some of these six-pack rings may ultimately in the environment because the waste collection is not perfect. And then our soil and water system may get polluted with six-pack rings. And eventually, that's not necessarily bad. It starts being bad when, for example, these things happen, like entanglement of wildlife in six-pack rings. And when a tourist comes across this poor turtle that's entangled in a six-pack ring, he takes a picture, and that then triggers a whole chain of events in society, because the public is is outraged by the fact that these adverse effects occur in the environment, and that may then be a reason to take measures to intervene in this chain. For example, to start cleaning up the water, or to develop alternative packaging materials, as already happened a lot, for example, in Europe, where you almost cannot find a six-pack ring anymore in the shops. It has all been replaced by paper packaging. Or you can also eventually try to change the behavior of people. But before you can take measures, this link between actually awareness and measures, public outcry and measures, it requires some steps. It requires that people are aware, it requires knowledge, it requires governance structure, and it requires resources. Otherwise, we cannot simply take the measures. And I put your knowledge in red because I think that's what we basically try to do. We try to integrate all the available knowledge in our models to make predictions. So modeling is basically important to do some kind of scenario study. So what happens if I intervene in this way? What will happen with the levels of plastics in the environment? So this example is for six-pack rings, but it applies just as well to microplastics. More in general, we say there is this dip search, so that you have the drivers in society, the pressures, the human activities that impact the soil and the water, so the state, and eventually impact biota, which then triggers the responses. And what we basically try to do as modelers, we particularly try to model this upper chain of drivers, pressure, state, and impact. You can also tell that in another way. We try to model the source to effect chain, starting at human activities and ending at the adverse effect in the environment. One practical area, one example where we do that, for example, is risk assessment. And just to illustrate that, if you would map chemical risk assessment in this chain, you would say, okay, what we do typically in chemical risk assessment is based on what we know about emission sources. We try to predict, for example, the concentration people or organisms in the environment are being exposed to. We call that a PEC, a predicted environmental concentration. And on the other hand, we try to model the effects, and based on our prediction models, we try to predict a no effect concentration, a PNEC it's called. And based on a comparison of the PEC and the PNEC, we get an impression of the risk. So modeling is important. Model is important to do scenario studies. Modeling is important to assess the risks of microplastics. So where do we stand actually when it comes to modeling microplastics? Because it has been, little is known about microplastics, but some activities have been going on, and I just want to go try to give a brief overview of where we stand, and also want to give some examples of what we did with our group at the Open University. So first, I would like to focus on the sources, and I just took two pictures from the web, trying to show the main sources of microplastics in the oceans. And these are actually quite recent pictures, giving a quite accurate image of the sources. But you might also know that when this microplastic issue came on the political agenda, at first we were all talking about cosmetics. But when you actually look at these pictures, you will see that personal care products or cosmetics are in general considered quite a small, responsible for quite a small part of the microplastics in our oceans. There are other sources that are much more important, and one of these sources, especially the wear from car tires, has been quite undiscovered until recently. And it was first addressed, I think, in a Norwegian report, but it's nice. We also made a contribution there, and one of our students, Peter Jan Kohle from our university, started with a simple literature review, very small for his master's study, and he wrote a small part about it, and he started calculating, okay, how much of a tire wears actually well over its lifetime. then he started calculating the number of cars, and he said, "Look, it must be an amazing amount ultimately reaching the environment." And based on these simple calculations, he ultimately first published a small paper in a Dutch magazine that was picked up by a Dutch newspaper, and eventually that resulted in a review that was published in 2017 on tire wear in the environment and the different routes and the different amounts, and making clear that this can actually be an enormous source of microplastics in the environment, responsible for up to 30, maybe some places 50% of the microplastics in our oceans. The big problem, of course, with microplastics, at least from tire wear, is that the detection is particularly difficult because it's black. And it's not easy to detect. Anyway, this is a nice example, I think, where that shows that also using logical reasoning and using modeling principles, you can discover quite innovative or make quite interesting discoveries. And a second example from our studies is a study from Jike van Wijnen. She did a PhD at the Open University, and she tried to do some integrative modeling of different sources to distinguish basically how much each source contributes to the loads in our oceans, especially when it comes to contributions from rivers. So she predicted concentrations at the mouth of rivers and looked back at, okay, what causes these? And she found, the results she found, of course, this is modeling, so I think these should be complemented with measurements, but the results are quite interesting in the sense that our models predict that about 80% of the microplastics at the mouth of our rivers comes from the degradation of macroplastics. About a little bit less than 10% comes from textiles and from tire wear, and only a very, very small fraction, probably less than 1%, come from cosmetics. So this is just an example of how a modeling study can contribute to kind of prioritizing different sources of microplastics in the environment. And that's important, I think, for risk managers, because they want to know which sources should we tackle first when we start taking measures. But of course, as said, it should not only be modeling, it should also be measuring and attributing the different particles to different sources based on measurement is also very important for this. When it comes to modeling the fate of microplastics in water, we have a lot of experience with modeling chemical compounds in water, but particles are, of course, a bit difficult. But at first, when you think about it, probably the fate is mainly governed by advective transport, so transport with the water flow. Although there are a lot of a variety of shapes and specific masses of microplastics, I think most knowledge is known. So we can model water flow. We also can model small particles in the water. What makes it difficult, I think, is that the properties of microplastics can change over time. There's sedimentation and resuspension, of course, but there's fouling. So actually bacteria that stick to the microplastics and can change then the size of the microplastic. There's fragmentation and degradation, which the falling apart of microplastics, which or larger plastic particles. And there is sorption and desorption of chemicals. And particularly this fragmentation and degradation and this fouling, which can influence sedimentation and resuspension, and that's where you can also influence advective transport. This is what makes it difficult at the moment to reliably model the fate of microplastics in surface waters and in rivers, I think, and also in oceans, by the way, because there the same processes, of course, take place. So modeling actually the fate of microplastics in surface water and in seas can still be quite a challenge. When it comes to land, and especially the transportation from the land to the water, to the rivers or to the to the oceans. There are also still some challenges where I think little has been happening over time. For example, if plastic is present on land, it can be mobilized by, for example, by the wind, and in that way be transported to rivers or the oceans, or it can be transported by precipitation. If it rains a lot, there can be runoff, and the runoff can mobilize the plastic and take it to the rivers and the oceans. These processes actually have been little modeled yet in our models. The first attempt I'm aware of actually, is from Meyer et al. This is a pre-publication yet. You can find on the interweb, so it's not yet a formal publication, but this was one of the first publications that I came across that actually explicitly tried the mobilization by wind and by precipitation, and from there the transportation to rivers and oceans. And I think that's a very important aspect that we should include in our modeling efforts. Then finally, exposure to humans. So humans, if you want to know how humans are exposed, you don't only have to look at the environment, but also in all kinds of different contact media. One of the contact media where we look at Open University is contact with food, especially seafood. And from that, I want to give a few examples or one example of a study done by Ineke Hantoro. This is a cooperation together with Sugiyaprana University in Indonesia, on the island of Java, in the city of Semarang. And Semarang, I will in a minute show a map and show where it's located. But there it's a town located at the border of the sea, at the edge of the sea. So there's a lot of seafood consumption. And we try to estimate how much the microplastics intake by inhabitants from Semarang, based on asking people how much seafood they consume. So we do a food frequency questionnaire, asking how often and what amounts of seafood they eat, as well as a 24-hour seafood recall diary to estimate the amount of seafood consumed. And of course, we try to measure the microplastic levels in the different types of seafood. And for this latter study, we go to, here's the map of Java Island of Indonesia. Here's the city of Semarang. And basically what we did, we sample 3 types of seafood in the coast that we catch ourselves. So milkfish, cockles, and green mussels. And we also explored 2 types of seafood that was taken from food markets in Semarang, so crab and shrimp. And we analyzed these for microplastics contents. So just to give you an impression of the studies, you see here on the left the milkfish ponds. So Semarang actually is known in Indonesia for its milkfish production. It's really a traditional food product there. The cockles are more consumed by the poorer people of Indonesia, and there's a picture from a fish market. Here, just some preliminary results, which I personally think is very interesting. So these results we obtained are now being analyzed and will shortly be published. But this is just to give you an impression of the end results we produce, showing intake of microplastics from different categories of seafood for different age categories of people. And one of the nicest things I find from this picture is that you actually see quite a difference between old people and the other groups. So old people consume more fish and therefore have a larger microplastics intake from the consumption of fish, whereas the younger groups consume more bivalves, and also the levels of bivalves are levels of microplastics in bivalves are higher, and therefore have a larger fraction caused by this. So this kind of illustrates that also the habits in the population in this case determine the exposure to the microplastics. It's not only the microplastic levels that's important. Okay, then moving from the exposure to effects. As said, I'm not really an expert in this area in the sense that I'm an experimental toxicologist, but I think I want to make some remarks about the little things I know about toxicity. And I think we can distinguish physical effects from toxic effects. So physical effects are, for example, if an organism would consume so many microplastic particles or plastic particles that it obstructs their the gastrointestinal tract and that it eventually dies. And for example, this picture there for this dead seabird with all the plastic in its stomach, and it simply starves because it doesn't eat anymore or because there's the tract is obstructed. Then of course, another physical effect more from microplastic can also be that these microplastics stick to the surface of your gastrointestinal tract, of your stomach, and then they can trigger infections, inflammations. That's also been reported. I just took one example of Chow et al. Here, picture, to illustrate. The big question is, of course, and that's a lot in debate at the moment, is these first two examples are more what I call physical effects, but whether there are also toxicological effects, and especially whether microplastics are translocated from what I still call outside of the body, which your gastrointestinal tract is actually still outside your body, and whether that passes the membranes and passes your barrier and actually has been transported to the inside of your body, your systemic circulation, and can end up in other organs like your liver. And for that, I think the particles need to be very, very small, in the below micrometer size. And of course, then substances that have absorbed to the particles, either additives or contaminants from the environment, can desorb again. Although I'm not sure whether the amounts that are of chemicals being transported into your body will really be significant when you would compare it to what's already out there. There's a lot of discussion going on in the scientific literature. The same more applies, I think, when it comes to the small particles, the nanoparticles that actually have been shown that they can be translocated. For example, in a recent study of the salsa that was reported in scientific reports. Actually, they convincingly showed for zebrafish, I think that was, that very small particles can end up in the liver, but they don't show any inflammation. So it is quite, there's a huge discussion at the moment going on actually about the toxic effects and whether these toxic effects are really relevant. When you would like to know more about, for example, whether the small particles can actually be taken up into your body, I would suggest to read this review of Wright and Kelly in E17 from 2017 that very well explained the different transport processes that can take place for microplastics over, for example, the lung epithelium or the gut epithelium. So. Then when it comes to specific toxic effects, I think the most likely effect, in my opinion, that microplastics could trigger inside your body once taken up is probably immunotoxicity. And there are more and more indications that this can happen. And for example, a recent example is here, the study of Sendra, also from scientific reports, that studied the effects of microplastics on hematocytes of the Mediterranean, Mediterranean mussel. And they showed, particularly in one type of cells, the R1 type, which is this left column here, that there can be quite convincing that there can be effects on the immunotoxicity. And that's not amazing, I think, if a strange particle enters your body, that your body responds by trying to remove these. But I also have I must say a bit of a problem with all these toxicity studies, and I try to summarize that here, because most toxicity studies are actually done using quite high exposure levels with microplastics, which are much higher actually than the exposure levels generally found in the environment. And there's of course also a kind of a mismatch between the particles that we can actually measure in the environment, which, as we have shown, are often larger than 50 to 100 micrometers. And if you are very good, maybe you can go up to 10 or 1 micrometer, but much smaller you won't come when it comes to environmental exposure. While a lot of toxicity experiments are being done with very small particles, more in the nano range. And then again, the toxicity studies are often done with particles with a uniform regular shape, while the particles that we actually find in the environment have a very irregular, variable shape. So it says here variable regular, but I mean variable irregular shape in this table. So basically, my impression is that there's a lot of debate about toxic effects, but the toxic effects have definitely not been convincingly shown to actually take place in the environment. And I don't expect over the next 10 years that toxicity studies will provide a clear answer to the question, what is a safe level for microplastics? Simply because the diversity is so enormous and it's a very complex problem. That triggers, of course, a question: what is an alternative for impetus for regulation? And I personally think that we might not want to wait for a toxicity to be proven as a toxic effect of microplastics. Maybe the omnipresence of microplastics in the environment itself is enough reason to take action, because we know from experience that if we change our abiotic environment by bringing a lot of products there that don't belong there, that also the biotic component will respond to that. So by changing our abiotic environment, we'll also change our biotic, our ecosystem, basically. And on the other side, there's also this socioeconomic impact of microplastics, especially from, for example, the media reporting on microplastics in our seafood. And people get scared that they get pollution. They're saying we're eating plastic fish. And they may start avoiding seafood. And that can have an enormous economic impact on this branch of industry. So one of the things that we proposed is maybe toxicity should not be the first reason to take action, but we might set food safety or action levels for microplastics in seafood, not so much based on toxicological considerations, but mainly based on considerations like, okay, this doesn't belong in the environment, and we should avoid for consumers that they think they're eating plastics. Okay, that brings me to the last topic, interventions. I think there are a lot of interventions possible, and a lot of people are working on interventions. A very important one is the development of degradable plastics as replacement of conventional plastics. But there are a lot of tricky sides on degradable plastics. First of all, they should, of course, not trigger microplastic formation, right? You can have a degradable plastics, but if the end product is a microplastic, you should not do that, because that will make the problem only bigger. And I think if it would result in the fact that degradable plastics, that people start thinking, oh, it's degradable, so we can throw it away, then there can also be a lot of indirect effect from degradable plastics, because then, of course, still to break it down, it takes energy, it takes oxygen, and that may also lead to oxygen depletion in surface water, for example. So degradable plastics, it's a good idea, but it may need not be the perfect solution for all situations, definitely. Replacement of plastics by other products might be a better solution in many cases, I think. Behavioral change could be an option, so that people don't throw things away anymore, which is, of course, important, but which is also very, very complex to realize in practice. filtering, for example, the effluent of washing machines, but also at wastewater treatment, so decentralized or centralized, improved waste collection and recycling. These are all options that a lot of people work on. What is a bit missing, I think, in my opinion, is that these solutions and these intervention options are all very interesting, but although a lot of people working on the technical elaboration of these techniques and making it technically perfect, relatively little attention is being paid to how you integrate these different intervention options into the production process of articles. And for that, I would plea for a more integrative approach, looking at production processes of plastics and see how you can change these. So not only the production, but the whole from the raw material to the waste phase, map what happens to these plastics and look at the different options where you can intervene. You can capture this under the name supply chain management. So try to map the supply chain of plastics goods, try to identify the locations in the supply chain where the microplastics get formed and leak to the environment, and then look at these different intervention options like biodegradable plastics or product alternatives, and which measures and interventions can be best introduced. And I think we should really work on this link of modeling actually the use of plastics in society with the leakage to the environment to solve the problem. A very promising study or PhD report, one of the best I've read over the last 10 years, I think, is a study from Karecki et al. I wholeheartedly recommend you to read it, which is, and this is just one picture, one example from this study, where she mapped macroplastic flows around littering in Switzerland, and also tried to predict concentrations in different locations in Switzerland. It's really a very, very good study. I want to conclude. My main message is here. I think quantification of the distributions of different sources of microplastics is very crucial, and we have to improve there. Of course, it varies from location to location and may also vary in time, but we need to have a grip on which sources are contributing most to the microplastic loads of our oceans. Regarding fate, I think understanding degradation and fragmentation of plastics is definitely crucial. Regarding effects, I think at the moment there's an enormous gap between the toxicity studies being performed and actually the requirements to assess the risk. And this gap will not be closed over the next 10 years, I think. And therefore, I think the management impetus should not come from the results of toxicity studies, but from other considerations. When it comes to interventions, I think there should be more focus on implementations of interventions in practical context, namely supply chains. And finally, I think models are helpful to keep the big picture in mind. So that concludes my presentation. I would like to thank my colleagues at the Open University, particularly Pieter Jan Kolen, Ineke Hantoro, and Jikke van Wijnen, from which I showed some results. And of course, also my colleagues, Antje Leur, Frank Belgen, and Harold Krikke. And with that, if you have any questions, I think we now move to the question and answering session. Thank you for your attention.
Thank you so much, Ad, for this excellent presentation that gives an important view about the current knowledge on microplastic research. So, yes, now it's time for discussion and Q&A. I think we have some questions on the Q&A section. I see that you already answered some of them, but I think that we can answer them again in this section because maybe other people may be interested in knowing these answers. So first, there is a question from Anouk Dont that says that he asked if some of the any of the speakers found an effect of water depth or distance to sources like river, city, or shore on the microplastic concentration in sediments. Means if according to the depth or the distance to the source, if you have found in your study some effect on the microplastic concentration in sediments. Maybe this, I would say that maybe it's a question more for Gilberto. Do you have any idea about this?
Well, I found a paper which was pretty cited recently. It's from 2016. It is a modeling one actually, but it is to reconstruct the fate in the wall section of a river. So it can be good to find the final sink in sediment or water. And it's from Nitset and other authors. It should be on, I don't remember actually. I can try to find out on the web and send a link there. It will be faster, I guess.
Maybe I can make a short remark. We did a review on microplastics in seafoods and then also looked at the correlation with microplastics in the surface water and in sediments. And, we found a very generic result, not with water depth, but of course with if there's more human activity, you generally find, of course, higher microplastic levels, but that's quite generic. So, and I think it would also always depend, of course, on the flow directions, on in the currents in the sea, etc., etc. So there's no one fits all answer to this question.
Yes, in some study we did some years ago with fish stomachs in the coast of Spain, we also found more microplastics in fish that were close to bigger cities like Barcelona in the Mediterranean than in places like Mallorca Islands. So definitely it seems that there is some effect, at least of the closeness to the sources of plastics. Yeah, then Elvis Shu from Denmark already asked Gilberto that 87% of the fishes contain microplastics in your study, but he asked in what tissues? So
well, I already answered actually, but I did not follow directly that study, but it was found in stomach tissues anyway.
Yeah. And also, Gilberto, can you also Francesco Bregoli asked you a question that about he says freshwater network is the pathway of plastics from lands to oceans, but there are not enough studies. We should push more on this topic, he says. And his question is, how did you sample in Lake Como, and why do you have a concentration of an order of magnitude less in respect to close similar lakes?
We measure through different transects with a fixed distance from the coast to avoid any kind of bias. And actually, we didn't find a specific trend to figure out how it is less polluted than other lakes, because actually Lake Garda, which I use with as a comparison, was sampled in the same way. So you can say it's not a bias due to sampling, for sure. Probably we have just fewer sources close to this lake instead of other European lakes.
Yeah, thanks. Then we have a question from Inek Hantoro to José Manuel. He says that regarding, she said regarding polymer identification using FTIR, can you recommend a lower limit score to be able to ensure that the polymer from the sample matches the existing database?
Yes, this is a typical question. Well, I have the mask because one of the collaborators is here, so don't worry. I am not afraid of you because of the internet. So the virus, fortunately, is not going through the internet. Yeah, about the score. Okay, the first idea is, in general, with the software, with infrared software, you will obtain a collection of matches, let's say 10, which are typical. And in general, they are ordered from the highest to the lowest match, let's say from 0.7 to 0.3. In principle, you can discard the smallest one. So 0.2, 0.3 in general means that the match is really, really bad. However, in many cases, you can obtain a large score, let's say 0.7, because of the coincidence of the main bands of the spectra, but not of the minor bands. And in these particular cases for polymers, in general, you are more interested on the small bands than in the largest bands, because the larger bands could be typically from OH, from water, from humidity, and from OH groups, hydroxyl groups that enter into the polymer. So don't trust only on the number. Visualize all the matches, focus on the fingerprint region, which is from 2000 centimeters to 600 more or less, so the right part of the spectrum. And if you find that the shape of the spectra are matched, then you can be quite confident. Do not focus just on the number, because this could be misleading, and don't think that a very, very good match on the right hand of the spectra are enough, because there, in many cases, the matches are good because of OH. If you have OH, if you have water, humidity, etc., you will have a very big band there, and you can think, oh, wait, this is a perfect match. So your problem will always be at the fingerprint region, which is the region at the right of the spectrum. So there you have to take all the possibilities, discard those that clearly are not matching because they are different, you cannot find the bands, or you can see different ratios, and then from the most probable situations, then this is where you have to concentrate. And also you need to take into account weathering, which is something which has not been addressed quite a lot in literature. So it's not a question of the number, it's a question of visualizing the match, visualizing that this spectrum could be this one.
If I can add something to this question, José Manuel, how this affects the standardization of the technique?
To the general protocols. Yeah, this is a big problem because it's very simple to say, if you obtain a score of 0.6, let's go ahead. The problem is that you can standardize the experimental work up to this point, because there you need to spend some time yourself in front of the screen, and you have to say, this is not good enough for me, or this is good, or this seems to be good, etc. So you could standardize everything up to this point, because this is the point where you need the scientific reasoning. Because this happened twice, here the collaborator, which is Adrian, is suffering the problem. You could have very good matches because you are considering the left-hand side of the spectrum. In fact, now he is considering only parts of the spectra to make the searches through the databases. So it's not possible, I think at least, it's not possible to standardize everything. You can standardize the measurement, you can standardize sample treatment. When you have to arrive to this point, you need to compare, and you know that comparison can only be by humans.
Yeah. Thanks. Then we have a question from Nora Exposito to Ragas. She says, Dr. Ragas, how can we extrapolate the microplastics concentrations found in seafood to toxicological studies?
Yeah, thank you for that question. It's a very good question in the sense that we are also very much struggling with this question. So we're actually measuring concentrations of microplastics in seafood, and when we started out, we had the idea, okay, then we're going to predict the risk, but at the moment it's simply not possible, I think, to actually predict risks in terms of risk of getting sick or risk of dying from microplastics or a risk of inflammation, because simply not enough results have been obtained there, and that's exactly what I meant with also the mismatch in the sense that we measure quite big microplastics, so in the sense bigger generally than 40, 50 microns, while toxicity studies often focus on the smaller particles. So it's really difficult to translate these exposure concentrations that we measure in seafood to toxicological risk. That's really a challenge at the moment. We're going to look into it in more detail over the next 2 years, but I don't expect to find an easy answer there.
Yeah, as many other challenges we have for the next years, you know. Yeah. Then we have a question from Hakan Selebi that asks which methods are used for microplastic measurements in terms of environmental engineering, and if the experiment procedure is available.
I am not an expertise on engineering, but I have seen that people is using metallic mesh to get rid of the microplastics in the wastewater treatment plants, for instance. If this is what he means, I don't know.
So other question is from Marie-Christine Desjean. Recycled plastic is known to contain, to contain more toxic molecules issued from catalysts because of manufacturing transformation processes. How do you cope with this question?
Maybe I can make an attempt, but I don't know the exact levels. But I know one study we did in Radboud University, a modeling study, and that didn't focus on additives, but on absorption of environmental contaminants to microplastics. What we basically try to quantify is compare what's the maximum load of contaminants, of chemicals that absorbs to microplastics, and in that way could enter an organism, and compare that to what's natural absorbed through just absorption processes of the chemical. And our modeling study at least showed that this fraction that is transported through microplastics is actually quite small compared to what's directly absorbed in the organism. So for at least the transport of environmental contaminants, I personally think microplastics do not really play a relevant role. But it may be different from the additives, so that are actually enclosed in the microplastics, especially if the microplastics would fall apart and they desorb. But I'm not aware of a lot of studies going on there, at least not at environmentally relevant level. So you know studies where they put a lot of PAHs or something on the microplastics and then expose the organism, and they show, okay, the PAHs are transported into the organism. That's of course true, but in reality, these levels of PAHs will never be in the environment and be transported into the organism. So again, there's this mismatch of toxicity studies and using high levels with what's really happening in the environment.
Yeah. Then Adam Greenwood asked what efforts are being made to quantify microplastics within the first meter of the soil? Which methods show promising results? So I guess similar ones to those of sediments, but do you have any idea about this question? Which methods are promising for measuring microplastics in the first meter of the soil?
As far as I know, they are using the same methods as for sediments. They need to take a typically graph sample, and they make some kind of separation, and then they float the microplastics in many cases. And then once you have floated the microplastics, you proceed as in sediments. You have to filter, you have to clean, and you have to go to the measurement system, typically infrared.
Some of the methods that you already explained during your presentation.
Yeah. I have a question from Pietro Giucciardi about the advantages and disadvantages of infrared or Raman. Well, the first question is about money. If you are going to perform monitoring studies or to measure quite a lot of samples, you need for sure infrared, classical infrared spectrometry, and you don't have to spend a lot of money. If you want to measure the same samples using only Raman, Raman is prepared to go particle by particle. So probably the battlefield could be infrared spectroscopy, classical infrared spectroscopy, combined with microscope. And if you want to study very, very small particles, let's say lower than 1 micron, then you have to go to Raman spectrometry. In principle, there is an optical limitation to measure by infrared spectroscopy, which is, if I remember correctly, 10 microns or something like that. This is like the physical limit in general. If you have to go down from 1 micron, you need to go to Raman spectrometry. But remember that Raman is particle after particle, and you could have an experimental problem, which is the fluorescence of polymers. In Raman, you have to use lasers to excite the sample, the particle in this case. And by this big excitation, typically you will have fluorescent signals instead of the infrared spectrum. So you will have some problem to get the correct spectrum. You can, if you want to avoid the problem, your system needs to have 3 or 4 different lasers, and you have to make tests with the 3 lasers and see which one is much better, which with one you can avoid fluorescence. So the answer is, if you will have a lot of samples, you need a battlefield, and the battlefield is usual infrared spectroscopy. If you want to focus on very, very small particles, and if you are ready to analyze one particle after one particle, then Raman could be useful. And remember that Raman is really expensive.
Yeah, thanks. Thanks, José Manuel. So, yeah, we still have some questions, but I think that we have to close the webinar now. So, yeah, thank you very much. I think it was very, very interesting. Again, I'd like to thank Gilberto and José Manuel for their excellent presentations. Also, all attendees for their questions. And also, I would like to thank Elena, Natasha, and the Toxics team for organizing and supporting this webinar. So thanks all for your attention. Take care, stay healthy, and hope to have a nice 2021. Elena?
So yeah, before we go, I would also like to thank you, Juan, for chairing this session, and all the 3 speakers for your interesting presentations. As mentioned in the chat, certificates of attendance will be available on demand to webinars@mdpi.com. And I hope you stay fine, take care, and see you soon in other events. Bye-bye.
Bye-bye. Thank you.
Bye.